Okay, so where were we again? Yes. So we in the last video we developed this idea that the conformational change induced by a molecule when it binds to a ligand binding site of a receptor will depend upon the molecular structure, how it fits into the receptor, and what parts of the receptor it interacts with and how it distorts the shape of the receptor. Hopefully that should be pretty kind of obvious now that we've looked at it. Now in this video I want to look at LSD. We haven't really looked at LSD, and LSD is kind of a little bit special, so we should spend a few minutes with LSD at least. And then I want to introduce two really important ideas in modern pharmacology based upon this principle that we discussed in the last video. And these are, I'll tell you later. I'll tell you in a minute. But they're really important. And they relate to this idea that the conformational change of the receptor depends upon the molecule and how it interacts with the receptor. But first, let's spend a little bit of time with lysergic acid diethylamide. I don't think we've really looked at his structure yet, so let's do that. Okay, so here we have a diagram showing LSD. Now, LSD is a beautiful and rather complex structure. So we'll redraw it here in a slightly more familiar form. So LSD, it's a tryptamine. So we start with the phenyl, which we then complete to form the indole ring, as usual. Then we add our two carbons, 1, 2, and the nitrogen, in fact, even. And there's even a methyl group here. So this is starting to look, well, it's almost like DMT, right? It's a tryptamine. It has the tryptamine chain. However, what's different about LSD is that you have another carbon that kind of then loops round. So you have this 6-membered ring here, and then this carbon here then connects down here. So you've actually got this 6, 5, 6, 6 kind of stacked ring structure, and there's even a double bond actually between there. Then you've also got this additional structure here. So this is called, this actually is lysergic acid. However, when you have a nitrogen here, you've got what's called an amide. This is why it's called lysergic acid amide, or type of amide. That is in fact lysergic acid amide, LSA, which you will find in morning glory seeds. But here, and that's a natural compound. But here we have the semi-synthetic compound, which contains 2 carbons here, 2 carbons here. These are called ethyl groups. And so we have 2 of them. So we've got diethylamide, lysergic acid diethylamide, lysergic acid diethylamide, LSD. Now this is obviously a far more complicated structure, should I say, compared to serotonin or DMT or even psilocin, right? It's got much more structure to it. This kind of chain on the tryptamine is kind of held in place, kind of locked in place by this ring structure. You've got this double bond here which can interact with the receptor. You've got this double bond oxygen. You've got a bit of a negative charge on the oxygen. You've perhaps got a positive charge, a little bit of positive charge on the nitrogen. You've got this ethyl groups here can interact with the receptor. There's lots of ways that there's the potential ways for the LSD molecule to interact with the receptor. Just by looking at it. Now, what we know is that LSD is an extremely potent psychedelic. What do I mean by potency? I simply mean how much of the drug is required to elicit the effect. With LSD, it's kind of 50 micrograms, 50 millionths of a gram, which compares with 50 milligrams for DMT, for example, or even up to kind of 400 or 500 milligrams for something like mescaline. So LSD is extremely potent. And it's always been something of a kind of a mystery of why LSD is so potent. It was always kind of reasonably clear that it seemed to have something to do with the complex structure of LSD, and that it seemed to perhaps to fit into the receptor in a very specific way. It has this very kind of rigid structure, kind of enforced by these kind of four rings, and then also something to do with this diethylamide group, perhaps interacting the receptor in a certain way and inducing a very specific tight interaction between the drug molecule and the receptor, which is perhaps responsible for its potency and its long-lasting effect. The fact that it lasts for 6, 7, 8, 9, 10, 11, 12 hours, as opposed to just maybe 3 or 4 hours with psilocybin, or 3 or 4 minutes with DMT. And this study actually, which I'll show you now, so this was a study which you can find here by Daniel Wacker et al., published in Cell, no less, which actually used X-ray crystallography as well as some modeling work to actually show, to visualize, using X-ray crystallography, the actual position of LSD within the ligand binding site, and actually showed that when LSD kind of slots into the ligand binding site, it causes a piece of the protein, the ligand binder site, to actually kind of fold over. They called this a lid. And this basically covers the LSD molecule and essentially locks it into the receptor site. And this is why LSD, once it's in the receptor site, it tends to stay there for a very long time. Other drugs, psilocin or DMT or serotonin, don't induce this change. They don't cause this folding over of part of the protein structure, which causes, which is referred to as this lid. So that's we kind of understand now what's special about LSD. So that's LSD, and we will return to LSD. But kind of so far hopefully now it kind of makes sense to you that when LSD or psilocin or DMT or mescaline binds into the 5-HT2A receptor, it induces its own kind of characteristic change in the receptor conformation. But of course, these are all agonists at the 5-HT2A receptor. DMT is an agonist, LSD is an agonist, psilocin is an agonist, serotonin is an agonist, but they all have different effects. They're all psychedelic, they all have different effects, apart from serotonin, of course, which is non-psychedelic. There are also other, as I said, there are other 5-HT2A agonists that are not psychedelic. So what's actually going on here? Originally, this was something of a mystery, because in kind of classical pharmacology, a receptor is either activated or it's not activated. It could be partially activated, which kind of explains some things, why some drugs are more potent than others, for example. But it doesn't really explain why a drug can activate a receptor and yet not be psychedelic, whereas other drugs that activate the same receptor are psychedelics. There's something different about the way they are activating the receptor, and we kind of understand this now. And in this video and into the next, we're going to talk about a really important concept called functional selectivity, as well as another important concept called receptor crosstalk. Now, briefly, and I will go into this in some detail, functional selectivity describes when two different drugs both activate a receptor, but they activate it in different ways. Now, what do I mean by that? What I mean by that is they activate different intracellular signaling pathways. Now, we have looked at, with the 5-HT2A receptor, one particular intracellular signaling pathway associated with the 5-HT2A receptor. So let's revisit that now and kind of remind ourselves of what we're actually talking about here. So we discussed this particular signaling path, this simplified signaling pathway for the 5-HT2A receptor. You have the ligand binds the 5-HT2A receptor, conformational change, interacts with this protein here called GQ. I'm going to emphasize GQ now. This is an important G protein. And then GQ activation leads to activation of phospholipase C, which leads to the production of DAG and IP3, which together in a kind of synergistic fashion activate protein kinase C, which indirectly through a number of other kinases activates ERK, this MAP kinase as it's called, which phosphorylates the potassium channel and inactivates it. And we showed how inactivation of these particular type of potassium channels leads to depolarization, the pushing of the membrane potential of the neuron towards the threshold potential, basically exciting the neuron, bringing it closer to the firing threshold potential. Okay, so that was one way of looking at activation of the 5-HT2A receptor, but I kind of lied to you a little bit. I've oversimplified things. And I want to explain why I oversimplified things a little bit. So this is kind of a bit of a paradox. So here are two drugs. One of them is one you're familiar with. One of them is LSD. LSD. And this one on the right is called Lyseride. Now, when you look at them, they look very, very similar, and they are very, very similar. In fact, the only real difference, if you look at them carefully, is that with Lyseride, you've got this extra nitrogen here, but that's basically the only difference between them. Now, they are both, and I'm going to write this because it's important, they are both 5-HT2A agonists. This one is, LSD is, and Lyseride is. LSD is psychedelic, of course. Lyseride is non-psychedelic. So why is that? So that was kind of something of a mystery in pharmacology for some time. We now understand that it has perhaps something to do with functional selectivity and receptor crosstalk. Now, that's not going to make any sense, but I want to spend the rest of the video talking about these two important ideas, and then in the next video we'll actually apply it to LSD and Lyseride and actually explain why, despite both being 5-HT2A agonists, only LSD is psychedelic, whereas Lyseride is non-psychedelic. So let's have a look at functional selectivity first. So this is our diagram from an earlier unit where we have basically antagonism versus agonism. So this is an antagonist. Doesn't induce any functional change in the receptor conformation when the antagonist binds, whereas the agonist here induces a conformational change in the intracellular domain of the receptor, which causes it to bind to signaling molecules, which activates them, which then activates other signaling molecules, and you get the start of an intracellular signaling pathway. Now, we're now going to kind of complicate this issue and realize there's actually more than one type of agonism. So this next diagram will illustrate what I mean by that. So we'll forget about antagonism completely now, where this is purely talking about agonism. So we've got here two different agonists. So we've got this blue one, this kind of ovoid shaped agonist here, and we've got this hexagonal one. These are both agonists. In other words, they both activate the receptor. However, what's different? Well, when the blue agonist binds to the receptor, it induces a conformational change in the receptor. We understand how that works now, which is transmitted to the intracellular domain. which causes it to interact with, in this case, this particular signaling molecule, which then interacts with this one, and we get this particular signaling pathway. This intracellular signaling pathway is activated, which ultimately leads to some kind of effect on the neuron. However, with the, let me just clear this off. But with in the second case, with the hexagonal agonist here, when it binds to the ligand binding site, it induces a different type of conformational change in the ligand binding site, and thus in the intracellular domain. And again, now you should understand how that can be possible. And it causes the intracellular domain to actually interact not with this signaling molecule, but with a completely different signaling molecule, signaling protein, perhaps an entirely different G protein, which then interacts with another different signaling protein here. So you get, again, the activation of a intracellular signaling pathway, but a different one. So the blue agonist activates this signaling pathway, whereas the yellow agonist activates this other intracellular signaling pathway. In other words, you have one receptor, but the actual signaling pathway that is activated by activation of the receptor depends upon the agonist. So this is functional selectivity. It's not simply a case that of a receptor activating a ligand activating a receptor, but the agonist, depending upon the agonist, different signaling pathways can be activated, which means that the overall effect on the neuron doesn't just depend upon which receptor is activated, but how that receptor is activated. This is an incredibly important and complexifying principle in pharmacology. You know, we no longer have to just worry about which receptors does the drug bind to, which receptors does it activate, but also we need to think about how does it activate these receptors. Okay, so that was functional selectivity. Now we're going to look at something called receptor crosstalk, which is slightly more complicated, but not overly complicated. So again, this is again based on the principle that when a ligand binds to a receptor, it induces a conformational change in the receptor. Okay, anyway, let's have a look here. So here we can see an agonist again. Okay, now a blue agonist here interacts with the receptor, induces a conformational change, which causes it to interact with these intracellular signaling molecules, and you activate this signaling pathway. Okay, fine. But notice something else that's happened here. As well as having this, as well as inducing this important change in the intracellular domain here, there also seems to be another change elsewhere in the receptor protein that doesn't seem to have anything to do with the signaling pathway. And that's entirely true. It doesn't have anything directly to do with the intracellular signaling pathway. It's just, you know, when the agonist binds to the receptor, it can cause a number of conformational changes. And the conformational change isn't necessarily restricted to a particular area of the intracellular domain, as in here, but can cause changes elsewhere. And these can actually be important. So let's look at another entirely different receptor. So here we can see a completely different receptor. It binds to an agonist shown here. Induces a conformational change in the receptor, which activates a particular signaling pathway in the normal way. However, again, we can see that there seems to be another change in the conformational structure of the receptor, that's unrelated to the intracellular signaling pathway. Now let's put these receptors together and see what happens. Well, it seems that when these both of these receptors are activated, the conformational change in each of these receptors that you can actually see here causes these receptors to actually, in a sense, stick to each other, to interact with each other. And this interaction only occurs when this conformational change is induced by the agonist, which is a little bit, perhaps a little bit confusing, but it's not difficult. So when neither of the agonists is bound to the receptor, then these receptors don't bind to each other. But when the agonists bind to the receptors, the conformational change, again unrelated to the signaling pathway, causes the receptors to kind of stick to each other. They kind of lock together in that situation. Now, what effect does this have? Well, perhaps no effect in some circumstances. But in some circumstances, when, let's say, receptor 1 here binds to receptor 2, it actually causes another conformational change in receptor 2, as shown here. Let me actually do this in a different color, in yellow, which actually makes receptor 2 less efficient in interacting with this yellow signaling protein. So it weakens this signaling effect of receptor 2. So receptor 1 is, in a sense, inhibiting receptor 2. So this is why we call it receptor crosstalk. Receptor 1 and 2 are speaking to each other by binding to each other. Receptor 1 binds to receptor 2, induces a change in the conformation of receptor 2, which weakens the signaling pathway that receptor 2, weakening the signaling pathway that receptor 2 activates. Whoa, okay, that's kind of this is very kind of really at the frontiers of modern kind of neuropharmacology here, because the ideas that receptors can speak to others in this way, binding to each other and actually affecting the way one receptor functions, is relatively new and actually quite controversial, particularly with psychedelic drugs. So in the next video, we'll come back to this idea. We'll hopefully kind of cement it and make it clear, and we'll actually talk about it specifically in relation to psychedelic drugs and how this receptor crosstalk and functional selectivity is important in distinguishing between agonists that are psychedelics, such as LSD and psilocin or DMT, and 5-HT2A agonists that are non-psychedelic, such as serotonin or Lysergic. So have a rest. See you in the next video. I'm kidding. However, if you do want to support the production of future courses, or you just want to leave a tip, then there are virtual tip boxes below. There's PayPal, Ko-fi, or Ko-fi, whatever it is, even a Bitcoin address if that is your thing. Minimally, please like and subscribe. It really helps me. And please follow me on Twitter and Instagram if you use them, Alien Insect. And I think that's about it. I hope you're enjoying the course. Thank you.